Yeah, so today I want to talk about the visualization of the composition of like microstructures in earth materials, and technique using is Atom Probe Tomography. I will mainly be showing like a couple of different research projects we were working on and others as well, and not so much go into detail about the different topics, as well to show what we can do on different types of microstructures. So the first thing is to say that the reason we are actually looking into this is that on that micro and nanometer scale where we operate when we're using Atom Probe Tomography, a lot of diffusion profiles and the transport of trace elements are controlled at that scale, which is one of the reasons that we are interested in this. But we can also derive a lot of information about, for instance, the geochronology of samples by looking at very detailed, for instance, lead compositions that are lost with larger analytical techniques. So first, a little bit about Atom Probe Tomography itself. There we go. So the way it works is that a specimen about 2 microns in length and 100 nanometers wide at the tip is placed in the machine, and then it's put under a high voltage that creates a very high electric field due to the very large tip. And atoms are field separated from the tip. They become positively ionized and then hit a position-sensitive detector. And so that records the time of flight as well as the order in which they come in, which then means that we have a March over charge based on the time of flight, and we can reconstruct it in 3D. And that basically brings us to the 3 main components of data that we can get from Atom Probe initially. So first of all, it is crystallographic information. Depending on the crystal structure, the detector will have a different hit map that's mainly visible in metals. In geological samples, that's often lost information. But as you can see from the first image here on the screen, you can then connect that to the poles that you would have from a diffraction pattern. Now, the second set of data set is the mass spectrum. So ions come in continuously. We hope always to get like single atoms that become ionized, but they often come as complexes with oxygen, which is very clear in rutile samples, where you can see Ti, TiO, TiO3, and they can become single positive ionized or double. And the third thing is that we can reconstruct that in a 3D image, but that will become a bit more apparent later on. But in these kinds of images, every single dot represents a single ion. So the tools we have are mainly, like after the analysis, the post-processing software. So often some groups use MATLAB. I think the main one used is IFS from Kamika that also develops the Atom Probe. And then we have several techniques that can go into the analysis of what we have actually, or the techniques, the analysis of the material that we've actually analyzed. So we can do mass spectral analysis, and we get a 3D reconstruction. But within those 3D reconstructions, we can start looking at clusters, we can create concentration profiles, we can make elemental maps, and we can isolate smaller and smaller regions of interest and do analysis on them. And so the microstructures that we've been mainly looking at for these kind of things are twin boundaries, low angle boundaries, fluid inclusions, and all kinds of clusters. And what I want to do in a moment is go through this work and then show how we can actually visualize that from the Atom Probe data. So first of all is that our workflow always goes from a higher scale to a smaller scale, and to make sense of what we actually see at the atomic scale, we first need to know what we're seeing at the larger scales. So it goes from field images to often electron microscopy, where we can then image where we can image the individual parts that we're looking for, and then use techniques like EBSD to visualize low angle boundaries, inclusions, use TEM to get specific information, and then get to ultimately Atom Probe Tomography, where we can see the individual ions and, for instance, the composition of a cluster like lead. So in order to do that, we follow this particular workflow all the way through. So we image it, we do the analysis, and then when we have found our region of interest and are done with the less destructive imaging, if we have something that we want to target, we mark that by putting a platinum dot onto it so that we can track it all the way through the sample preparation. Then following on from there, like with the TEM, we can use a focus ion beam to make a cut in the SEM around the sample that's placed onto a silicon stub and then milled by the ion beam into a very sharp needle. So that's basically the basics of what we're going from. So the first type of analysis like mainly has been doing of people looking at zircon. So this is a study from Peterman et al. In 2016 looking at discordant zircons, where they found that the core and the rim of the zircon didn't have the same lead composition. And so they selected a region from which they eventually made atom probe needles, and they found these dislocation loops embedded with lead inside these needles. And what we then can do with the software we have is that we can isolate these clusters, and in this case they have drawn a isoconcentration surface around it that can be isolated, and we can create an individual mass spectrum from those single envelopes around the inclusion. And the nice thing about that is that once we have such a very small volume with an own mass spectrum, that greatly increases the signal to noise ratio of our lead signal. And so we can actually then individually date these clusters. And as you can see from the Terra-Westenberg diagram here, that pretty much lines up with what's predicted from the LASRIM and the laser ablation studies they've done. So we did a similar study on a concordant rutile, and here you also see more of the 3D environment where you can actually look at the cluster orientation, everything in 3D. So this one is a sample from Australia that we knew had seen multiple high temperature events, but in laser ablation studies did not show any discordance. Yet still, we found all these kinds of clusters in there. And one technique we used to analyze these is to do a nearest neighbor distribution to see something about the processes which they formed. So because the lead comes from the uranium decay, that creates alpha recoil. And so if this is such an event, you would expect that there's a maximum amount of distance or minimum amount of distance that you can have based on that energy that releases. Now, since this doesn't fit, we assume that it's more in situ and it's in place of where the final uranium to lead transitions takes place. But we can, like before, we can isolate this material and then also start looking at the chemistry of these particular clusters. And so what we found in these is that besides the lead, we also see an increase in aluminium, we see an increase in chromium and vanadium. And so those proximity histograms are always measuring the concentration outside and inside a particular envelope, and that's perpendicular to that surface. Then on inclusions, so this is from the Boskedal in 2021, and this also nicely shows the correlation from what you can do with TEM and Atom Probe when you combine it. So they were looking at fluid inclusions in pyrites that have a dislocation pinned on them, and what they were able to show is the transport of elements from the fluid inclusion along the dislocation. One reason I've also included it here is that in terms of visualization, they created 2D profiles of the arsenic composition and made that into a 3D box, and then have drawn the position of the dislocation and the fluid inclusion inside it by, again, using an isoconcentration surface, this time about around 1 atomic % for arsenic and oxygen, as a metric for the position of the dislocation and the fluid inclusion. And we can then go on and do different measurements as well. So you can measure the concentration in these profiles. You can create a 1D concentration profile as they've done here. But this nicely shows how, to what extent you can actually image and what kind of images you can create. So to continue on into other microstructures, so we can look at twins. Basically, any boundary we can see and is coherent, we can target. And so this is a sample, again, from rutile from ultra-high temperature from the Napier complex. And so what we did here is that we looked at the dislocations via the TEM, and then try to, from the same boundary, also get the Atom Probe data. And so here you have a 3D render of these needles that shows the position of the boundary that's here highlighted by an OH group. And we've drawn an isosurface around that, which then shows the position of this. And what we can do with this data is then, for instance, measure the concentration above and below this boundary and inside it. And what we showed here is that there is a difference in how much every element is concentrated within it or not. Niobium, for example, is not really enriched with inside these boundaries, but iron and chromium are, which then has implications for trace element transport and so on. Another thing we've done here is use the similar 2D concentration profile as before to show that the uranium in these samples is actually not located inside the boundary, but has a profile above it. And here we also show that there is again a nice correlation between using Atom Probe and TEM, where you can see similar kinds of structures, both as enrichment in trace elements as in structure of the dislocations. So here we go in low angle boundaries, and low angle boundaries are a bit more complex than twins. They are less predictable. So here we made the specimens, and we also used TKD to actually check if inside our specimens we actually hit our boundary. So this is a ultra high pressure sample from Lago di Signana, and we place our markers, and we hit from the 6 2 that actually had that low angle boundary inside it. And when measuring that, you get these kind of profiles. So these are highly complex systems, but for instance, what you can do with Atom Probe is then start picking those individual parts apart. So we can see that calcium and iron are partially co-located, partially not. And we can also see that, for instance, the calcium, if you want to get into that, is in some of the branches along here, and so you can make a video in IVAS. And what you see here is that when we get along these entangled boundaries, you can see the iron peak is still high, but the calcium is only located in one. So this is all kind of information that you can extract from that image. Again, we make a proximity histogram that then shows more of an average of the full story. And we can also then do 1D concentration profiles to see what the differences are between the individual features within everything. And then I think the final thing I want to show is about phase boundaries and how to use the mass spectrum. So this again an ultra high temperature sample from a monazite inclusion in rutile. The monazite inclusions are around 500 nanometers wide. We did TEM images that show the whole world of complexity. And in this case, we also then made an Atom Probe needle out of this. We captured mainly the rim, but you can see different kinds of inclusions within this. And Atom Probe Tomography allows us to very specifically measure the concentration of these and visualize it. We can see that the boundary itself is denser at the bottom than at the top. So there's a difference in trace element distribution there. That's transitions towards dislocations that we can visualize here at the bottom, where you can see that the concentration of lead, silica, and aluminium is higher in these dislocations and also higher at the bottom and the top. And then the final part of this, and the last slide, is when you have such a data set, it creates a very, very massive, complex mass spectrum. But because we can isolate all these parts, for instance, with proximity histogram with isoconcentration surface, and we can then create individual interfaces, we are actually quite capable of picking apart all these this particular information quite well. And so we can show, for instance, that the lithium is not present in the rutile, but more in the monocyte at the interface. Calcium is only present in the interface. We can see lead is present at 2 different locations, and we can actually then measure, again, the lead isotopic concentration for potential geochronology. And that was it, and the references. And